everyone, Gayatri Premkumar here, and today we are going to discuss about the first major experiment, the one and the only major experiment, isolation of DNA from the plant material. So this video comprises of 7 different sections that includes the introduction of the experiment, aim and materials required, preparation of solution, what are the solutions required and what are the composition of the solution, and then the procedure, how you do the experiment, and observation, what you get out of the experiment, the end result, and what are the precautions to be followed while doing the experiment, and finally the viva question, the possible viva questions that can be asked in this experiment. The first section is the introduction. Like, what is the purpose of this experiment? Isolation of DNA from plant material. So first, what is DNA? You all know that the full form of DNA is deoxyribonucleic acid. So this is the full form of DNA, deoxyribonucleic acid, and we are isolating the DNA from any of the plant material. So what is the purpose? And this forms the basic of the biotechnology, whereas one of the application of biotechnology is recombinant DNA technology. What is this recombinant DNA technology? Where you cut or isolate DNA from one organism and introduce into the other organism. So this foreign DNA introduced into the host cell, and thereby you form a recombinant organism or recombinant DNA. So for the basic knowledge of this, so usually it is a higher level where the bacteria, yeast, or any other form of plant cell or animal cell, where you cut open or remove the DNA and introduce into the other organism. To have a basic knowledge about that, we take this experiment. So for students of 12th should have an idea about what is the application of biotechnology, how you get the DNA. For this purpose, this DNA is isolated from the plant material in a very simple procedure. So this forms the basis of the experiment, where we are isolating the DNA from the plant material. Most of the living organisms have DNA as the genetic material. An exception is seen in most of the viruses, where RNA remains as the genetic material. And the next one is about the aim and the requirements. So coming to the aim, so you write aim as to isolate DNA from the given plant material. So which kind of plant material can be used? So then we will talk about the requirements. So in coming back to requirements, where what all the things you require. So first we'll discuss about the aim. So when you talk about the aim, so you write to isolate DNA from the given plant material. So this will remain your aim of the experiment. This is this experiment is for 5 marks. The mark allocation for this experiment is 5 marks, and this is the one and the only major experiment in your biopracticals. So this is a confirmed experiment that will come for your biopracticals. So you should know what are the possibilities, what are the content present in the experiment. Then comes the requirement. When I'm talking about the requirement, that includes the apparatus or the glasswares, as well as the solutions that is required to complete the practicals. So what are the materials that you require? Can you guess? The first important apparatus that is required is the mortar and pestle. Like you have a mortar and a pestle ware. So with this you grind it. Okay, whatever the plant material is there, you grind in this mortar and pestle, and along with that you require other things, like you require test tubes, and then you require beakers, test tube stand, and all the other things you require for this isolation of DNA. Then we will discuss about the solutions. So what are the solutions that is required for doing this experiment? Now the next what we are going to see is the preparation of solutions that is required for the isolation of DNA. So what are the solutions that is required? The first thing is the detergent salt solution. In the name itself you can find out it is a combination of liquid detergent as well as the salt. So how do you prepare the salt solution? Either it is prepared and kept ready, or you are going to prepare. Anyhow, during the practical examination, you would have prepared it, or your teacher would have told you. But still, you should know how this detergent salt solution is prepared. It is prepared by taking 10 ml of the liquid detergent. Any of the washing liquid detergent is taken, and to that 10 gram of NaCl, that is sodium chloride, non-iodized sodium chloride is taken and added to 90 ml of water. I repeat, this detergent salt solution is prepared by taking 10 gram of non-iodized sodium chloride and 10 ml of liquid detergent to 90 ml of distilled water. And following that, the next thing what we require is the meat tenderizer. So it is available in the form of enzyme. So 5 gram of tenderizer is taken and added to 95 ml of water. Suppose if tenderizer is not available, what can be the substitute for that? So you can add or you can take either the juice of papaya or pineapple and filter through the muslin cloth, and that can be used as the meat tender instead of meat tenderizer. The next one is about NaCl. Along with that other two solutions, you also require 5% of NaCl. So how do you prepare this 5% of NaCl? It is dissolved in 5 gram of NaCl is taken and dissolved in 100 ml of distilled water. That gives the percentage 5% NaCl. The next is 95% ethanol, and again that is prepared already and kept ready, and but this ethanol should be used chilled ethanol. That is very important for precipitation of DNA. If you add, instead of adding normal ethanol, chilled ethanol will result in the precipitation of DNA, where the bottle itself is kept overnight and kept in the refrigerator. And one thing what we can do is during the examination time to maintain that chillness. So usually when each person is taking the bottle and using it, by the time the last person uses, the chillness of the ethanol may go down. So what we can do is either you can distribute in different beakers or test tubes and keep it in the ice cubes till the last person uses, so that the temperature will be maintained. So that you can follow. And then apart from these solutions that is prepared, you require one important important material. What is that? That is the plant material from which we are going to isolate the DNA. So what kind of plant material can be taken? So any kind of plant material, it could be either the papaya or green pea seeds or even the spinach leaves or banana, which is easily available. So any one of the plant material can be taken and smashed in the mortar and pestle to isolate the DNA. So this is about the preparation of solution. We have discussed about the aim, materials required, preparation of solution, and next we will go with the procedure for isolating the DNA from the given plant material. The next what we are going to see is about the procedure that is involved in the isolation of DNA. So what we are going to do is take 5 gram of the plant material. So as I mentioned already, the different plant materials, from that any material can be taken. 5 gram of any plant material is taken, and to that 10 ml of already we have prepared the detergent salt solution, no? So that will be taken and is grinded and smashed in the mortar and pestle. So you smash the plant material, and as you smash, you add this detergent salt solution, and whatever the extract you get, it is filtered through the muslin cloth. So the extract is filtered through the muslin cloth, and what are the extract, the filtrate you're getting, out of that you take only 10 ml of the filtrate, and to that you add 3 to 4 ml of tenderizer or papaya juice or pineapple juice is added, and then you have to swirl the test tube between your two hands. So what you do, you grind the plant material, add the detergent solution, filtered through the muslin cloth, and the filtrate what you get, to that you add the tenderizer, and the test tube is swirled between your two hands, and then you pour 10 ml of chilled ethanol, which I have told already. So you pour one the chilled ethanol along the sides of the test tube, and then you have to leave it for 3 minutes. 2 to 3 minutes you have to leave it. Then you can see that the DNA gets precipitated, and you can see white colored DNA precipitated. And what you can do now is, this DNA which has precipitated, you can take separately and keep it in a separate test tube containing either 5% NaCl or distilled water, and that you can show it to the examiner. Okay, either you can show it directly with that or with the 5% NaCl or the distilled water. So this remains the procedure for the isolation of DNA from the plant material. Next we will see what is observation. So what I have told you here in the procedure will remain the observation. So the white color precipitate of DNA that is formed will be the observation of this experiment, and next we will see the precautions to be followed and what are the possible viva questions that comes in this experiment. First part of this experimental description is about the observation. So what you will write in the observation area, that the precipitate of DNA, the DNA gets precipitated as white fine threads. So that is what you're going to show it to the examiner in the test tube. And then following that, so what are the precautionary measures that has to be followed while you're doing this experiment? The first thing is, whatever the plant material that you're taking should be washed completely to remove any kind of dust particle or any other particle present. After washing, you dry it and then you weigh 5 gram of that plant material. And then the glasswares what you are taking for the experiment should be clean and dry. It should not be wet along with the water. That may vary the experimental result. So you should take clean and dry glass apparatus, and whatever the chemicals and enzymes that you are using should be of standard quality, and the spectrophotometer. So after doing all these things, whatever the DNA that you're getting out of that, so that is measured using the spectrophotometer, which is instrument where which is an instrument where you measure or estimate the quantity of DNA using the light, amount of light. So amount of light intensity of that, you find out the or estimate the quantity of DNA. And then coming to the viva questions. So the viva questions might be asked in such a way that what is GMO? That is genetically modified organism, where you modify the gene of that particular organism by removing a part of DNA or adding a part of the new foreign DNA to that. And then detergent. What is the purpose of adding the detergent to this solutions? You have different preparation method, no? So why we are adding detergent? So it allows the membrane to dissolve the membrane and thereby release the DNA. So why we add meat tenderizer, either enzyme or it could be the papaya or pineapple juice? Why do we add that? Again, to remove any kind of protein that interfere with the precipitation of DNA. And last one, why do we add chilled ethanol? It is for precipitating DNA. So these are some of the possible viva questions. It might come any nook and corner of the experiment that connects along with the theory part also. So these are the possible like overall we have covered few areas of the question, but still as you learn you may come out with so many different information that relate with the experiment. Hope we have covered the aim, materials, procedure, observation, precautions and viva questions that relate with the isolation of DNA from the plant material. So hope you understood all this information. Thank you.